Hello, my name is Katie Munson, and I would like to welcome you to another video for MCDB 427, which is Molecular Biology at the University of Michigan. The figures in this presentation are from the book Molecular Biology, written by Robert F. Weaver, and in this video we will be discussing Figure 17.23, Principle of Toeprint Assay, Figure 17.24, Results of the Toeprint Assay, and Figure 17.25, the effect of competitor RNA on formation of the 48S complex. The diagram below gives a summary of the different factors and their roles in translation initiation in eukaryotes. It shows the major classes of initiation factors, and in this video we will be focusing on how the roles of eIF1 and eIF1a were determined, which are starred below. Note that all eukaryotic initiation factors begin with an E. Before we jump into the figures, let's review the two complexes involved in translation initiation of eukaryotes. Complex 1, also known as the pre-scan complex, occurs when the 40S ribosomal subunit forms at the cap but does not progress far, if at all. Complex 2, also known as the post-scan complex, occurs when the 40S has scanned downstream to the initiation codon and formed a stable complex. eIF1 causes only a modest, about 20%, stimulation of translation activity in vitro. Therefore, it was long thought to be dispensable. However, the genes encoding both eIF1 and eIF1a are essential for yeast viability. So, Tatiana Pestova and colleagues wanted to determine what roles these factors play in translation initiation. The toeprint assay is based on primer extension technique. It is used to locate the leading edge of the 40S ribosomal subunit as it is bound to an mRNA. Figure 17.23 demonstrates the toeprint assay and gives three experimental conditions. The first is a control in which you leave out the 40S ribosomal subunit so no complex can form between 40S ribosomal subunits and mRNA. The second involves 40S subunits, ATP, and all initiation factors except eIF1 and eIF1a. Complex 1 forms in the absence of eIF1 and eIF1a. The final condition involves 40S subunits, ATP, all initiation factors, including eIF1 and eIF1a. Complex 2 forms in the presence of eIF1 and eIF1a. Each of these conditions was mixed with a primer that binds downstream of the initiation codon on the mRNA. The primer was extended with labeled nucleotides and reverse transcriptase. Reverse transcriptase stops when it hits the leading edge of the 40S subunit. The length of the extended primer shows where the leading edge lies. Then, the primer extension products were electrophoresed to measure their sizes. With no 40S to block the reverse transcriptase, the primer is extended to the 5' end of the mRNA. When complex 1 forms, the primer is extended a long distance to the leading edge of the 40S particle. When complex 2 forms, the primer is extended only a short distance before it is blocked by the 40S subunit. The labels on the right correspond to full-length runoff extended primer, complex 1 and complex 2. For complex 1, the leading edge of the 40S particle under these circumstances was between positions plus 21 and plus 24 relative to the cap of the mRNA. This is about where we would expect it if the 40S subunit bound at the cap and did not begin scanning or scanned at most a short distance. For complex 2, the leading edge of the 40S subunit was between positions plus 15 and plus 17 relative to the A of the AUG initiation codon. This is about where we would expect it if the 40S particle was centered on the initiation codon. Lanes C, T, A, and G are the results of sequencing a DNA corresponding to the beta globin mRNA. They were included as markers to determine the exact positions of the leading edges of the 40S ribosomal particles in the complexes. The position of the initiation codon is given at the left. Also note that the runoff mRNA can be seen in each lane, which is due to the fact that even when all of the factors were added to form complexes, complexes did not form on every mRNA, and some of the mRNA was able to be completely reverse transcribed. Lane 1 shows the length of the mRNA alone with no complexes formed. Lane 2 has mRNA and 40S added, but no initiation factors or tRNA, so no complexes formed. Lane 3 has mRNA, 40S, all of the initiation factors, and met tRNA, so complex 1 was able to form. Lane 4 had all of the factors from lane 3, including a mixture of initiation factors obtained by salt-washed ribosomes. These factors can be washed off of ribosomes because they aren't bound tightly. The initiation factors that were collected were those proteins that could be precipitated by ammonium sulfate concentrations between 50 and 70%. This method is known as an ammonium sulfate precipitation, which is used for protein purification and separates proteins by altering their solubility and the presence of a high salt concentration. For this experiment, ribosomes were washed with ammonium sulfate to isolate those loosely bound initiation factors. This mixture of initiation factors could promote the formation of complex 2. Lane 5 includes all of the factors to make complex 1 and also adds eIF1. In this scenario, some complex 2 formed, but there was mostly runoff in complex 1. Individually, eIF1 had little ability to stimulate complex 2 formation. Lane 6 includes all of the factors to make complex 1 and also adds eIF1a. In this scenario, eIF1a had little ability to stimulate complex 2 formation. This lane includes all the factors to make complex 1 in addition to both eIF1 and eIF1a. These two factors together caused complex 2 to be formed almost exclusively. Looking at lane 8, complex 1 was allowed to form for 5 minutes, then eIF1 and eIF1a were added, and only complex 2 formed. Complex 1 was not stuck. Initiation factors could still convert it to complex 2. Based on these experiments, a few conclusions were made. First, eIF1 and eIF1a act synergistically to promote complex 2 formation. Second, complex 1 was not a dead end. Initiation factors could convert it to complex 2. Although the toeprint experiments led to some conclusions, they also led to some major questions. Did eIF1 and eIF1a convert complex 1 to complex 2 by simply causing the 40S subunit to scan further on the same mRNA? Or did these factors cause the 40S particle to dissociate from the mRNA and bind again to scan the initiation codon? Tatiana Pestova and colleagues sought to answer these questions by performing competition assays. Here is some background information to explain how the cold competitor functions in this assay. When there is no competitor added, if you pre-bind hot mRNA, but then the complex falls off and can rebind mRNA, it will only rebind to hot mRNA because there is no competitor present. However, in the presence of a cold competitor, if you pre-bind to a hot mRNA, but then the complex falls off and can rebind mRNA, the odds are that the complex will form on the cold mRNA and be invisible. For this assay, 32-phosphate labeled beta-globin mRNA was incubated with 40S ribosomal particles, plus the initiation factors, and a 15-fold excess of unlabeled competitor RNA in different combinations. The first condition, shown by the blue line, has all of the initiation factors added to form complex 2, including eIF1 and eIF1a, and does not add the competitor. If any dissociation and rebinding occurs in the absence of competitor, it will be on the same labeled mRNA. The next condition, shown by the green line, added eIF1, eIF1a, and competitor at time 0, along with the other factors. For this condition, no visible complex is seen because complex 2 formed on cold mRNA. The last condition, shown by the red line, include all of the factors to form complex 1, incubated the factors for 5 minutes, and then added eIF1 and eIF1a and competitor. For this condition, complex 1 was formed first on hot mRNA, but then there was a shift to complex 2, all on cold mRNA. After performing the incubations above, they separated the mixtures by sucrose gradient ultracentrifugation to detect the formation of stable 48S complexes, presumably equivalent to complex 2. Then, they detected radioactivity in each fraction by scintillation counting and plotted radioactivity in counts per minute. The 48S complexes that were detected were presumably equivalent to complex 2, involving 32-phosphate labeled mRNA, initiation factors, MET initiator tRNA charged with methionine, and 40S ribosomal subunits bound at the initiation codon of an mRNA. There was a radioactive peak of 48S complexes in the absence of competitor mRNA. all of the complexes that formed were only on labeled mRNA. Therefore, this shows you the size of complex 2, which we know because all of the factors were added to form complex 2. There was no radioactive peak of 48S complexes when they added the competitor mRNA at the beginning of the incubation, or when they added the competitor mRNA after complex 1 had formed for 5 minutes. For the green line, no visible complex is seen because complex 2 formed on cold mRNA and very little formed on hot mRNA, if at all. eIF1 and eIF1a and competitor were added at the same time as the other factors, and complex 2 was formed on the competitor mRNA instead of the labeled mRNA, because the competitor was in excess. This condition shows that with this amount of competitor, you don't really see much complex 2 on the radioactive mRNA. For the red line, recall that complex 1 was formed first on hot mRNA, but then there was a shift to complex 2, but all on cold mRNA, once eIF1 and eIF1a and competitor were added. This shows that eIF1 and eIF1a are able to disrupt complex 1. Based on this assay, several conclusions can be made. First, eIF1 and eIF1a did not simply allow 40S subunits in complex 1 to scan downstream and form complex 2 on the same labeled mRNA. These factors disrupted complex 1 on the labeled mRNA and forced a new complex to form on the excess unlabeled mRNA. Finally, eIF1 and eIF1a are not only essential for proper 48S complex formation, they also appear to disrupt improper complexes between 40S ribosomal subunits and mRNA. Thank you for watching, good luck with your studies, and go blue!